In 1989, the Exxon Valdez ran aground off the coast of Alaska and spilled 11 million gallons of crude oil into Prince William Sound. The response was enormous. 11,000 workers, 1,400 vessels, 58 aircraft, high-pressure hot water hoses blasting oil off 1,100 miles of coastline. And the single most effective tool in the entire operation was invisible to the naked eye. Bacteria. Wild, unengineered, competing with themselves and still outperforming every mechanical and chemical method deployed. That was the wild version. Effective, but incomplete, and 50 years later, still the best tool anyone has deployed. 18 years before the Valdez ran aground, scientists had already engineered a bacterium capable of consuming 2/3 of the hydrocarbons in an oil spill, 50 to 100 times faster than anything found in nature. It was patented, it worked, and it has never been used in a major oil spill. What happened between the laboratory and the ocean is a story about science, law, and who gets to decide when a discovery becomes a tool. To understand what Ananda Chakrabarti built, you have to understand what oil actually is and why cleaning it up is so hard. Crude oil is not a single substance, it behaves like hundreds of different problems at once. Some molecules evaporate within days of a spill. Others, the heavy polycyclic aromatic hydrocarbons, are dense, toxic, and extraordinarily persistent. They sink into sediments, coat feathers and fur, infiltrate food chains, and remain chemically stable for decades. The standard tools for oil spill cleanup, booms to contain the slick, skimmers to remove it from the surface, and chemical dispersants to break it into smaller droplets. Do not destroy the oil. They move it, dilute it, or break it into pieces small enough for something else to deal with. That something else is biology. Hydrocarbon degrading bacteria exist naturally in every ocean on Earth. They are specialists, organisms that have evolved. Over millions of years of exposure to natural oil seeps on the seafloor, to use petroleum hydrocarbons as their primary food source. When an oil spill floods their environment with food, their populations explode and they begin consuming the contamination. The problem is coordination. Natural oil-eating bacteria are not a single species working in concert. They are dozens of competing species, each optimized for a different type of hydrocarbon. In a real spill, they do not cooperate. They compete for nutrients, for space, for the specific fraction of the oil that each one prefers. The result is inefficiency. Different species work at cross purposes, and the most toxic, persistent fractions of the oil often go untouched because no single species is versatile enough to handle all of them. In 1971, a microbiologist working in a General Electric laboratory decided to fix that. Ananda Chakrabarti was born in 1938 in Sainthia, a small town in West Bengal, the youngest of 7 children. He earned a PhD in biochemistry in Calcutta, spent 6 years at the University of Illinois, and in 1971 joined General Electric's Research and Development Center. His first assignment was cow manure. He was soon redirected. General Electric had heard about the problem of oil-eating bacteria, specifically that 4 known species of oil-metabolizing Pseudomonas bacteria existed, each capable of degrading a different fraction of crude oil, but that they competed rather than cooperated when introduced into a spill together. The genes responsible for hydrocarbon degradation in each species were not in the bacterium's main chromosome. They were carried on plasmids, small circular loops of DNA that exist separately from the main genome and can be transferred between bacterial cells. Chakrabarti recognized this as an opportunity. If the degradation genes were on transferable plasmids, and if those plasmids could be combined into a single organism, the result would be a bacterium that could attack multiple hydrocarbon fractions simultaneously, without the competition, without the inefficiency. He spent months transferring plasmids between strains, combining the degradation capabilities of all 4 species into a single Pseudomonas cell. The problem was stability. Plasmids have a tendency to be lost during cell division, and after a few generations, the new combination would fall apart. Chakrabarti solved it by irradiating the organism with ultraviolet light after the plasmid transfers. The radiation caused the plasmids to cross-link with each other, fusing them into a stable, heritable structure that would not separate during replication. The result was a new organism, not found in nature, not a variant of anything that existed before. A bacterium that Chakrabarti called multiplasmid hydrocarbon degrading Pseudomonas, now known as Pseudomonas putida. Capable of digesting roughly 2/3 of the hydrocarbons in a typical oil spill, 50 to 100 times faster than the 4 natural strains working separately. He had engineered a solution to one of the hardest problems in environmental cleanup. Then he tried to patent it, and that is when things got complicated. In 1972, General Electric filed a patent application for Chakrabarti's bacterium with the United States Patent and Trademark Office. The examiner rejected it on a straightforward ground. Living organisms are not patentable. This was not a new position. The Patent Office had been refusing patents on living things since the 19th century, on the logic that nature's products belong to everyone. A bacterium, even a modified one, was a living organism. It could not be owned. General Electric appealed. The Board of Patent Appeals agreed with the examiner. General Electric appealed again to the Court of Customs and Patent Appeals. This time, they won. The court ruled that the fact that microorganisms are alive was, for patent purposes, without legal significance. The Patent Office appealed to the Supreme Court. By then, the case had become something much larger than one bacterium. General Electric was not fighting for the right to profit from an oil cleanup tool. They were fighting for the right to own the blueprint of life itself. The principle was that a living organism, if sufficiently modified by human hands, could be property. Every pharmaceutical company, every agricultural conglomerate, every researcher who had ever imagined turning biology into a product was watching. The case was argued on March 17, 1980, 8 years after the original application. Chakrabarti had long since moved on to other research. The bacterium he had built sat in limbo, while the legal system tried to answer a question it had never confronted before. Can you own life? On June 16, 1980, in a 5-4 ruling, the Supreme Court said, yes. Writing for the majority, Chief Justice Warren Burger held that Chakrabarti had not discovered a natural phenomenon. He had created something new. A bacterium with markedly different characteristics from any found in nature, produced through human ingenuity and research. The majority leaned on a phrase from the 1952 Patent Act's legislative history, that the law was intended to cover anything under the sun made by man. The 4 dissenting justices argued that Congress had never authorized the patenting of living organisms, and that the court was legislating from the bench on a question of profound ethical and practical consequence. They were right that the consequences were profound. But the majority prevailed. When the decision came down, the biotech industry celebrated, and for good reason. Diamond versus Chakrabarti opened the door to patents on genetically modified crops, transgenic animals, human gene sequences, monoclonal antibodies, and the entire commercial architecture of modern biotechnology. General Electric had fought 8 years to own one bacterium and ended up owning the legal blueprint for an industry. Its patent on the organism expired in the 1990s quietly, without ceremony, and without a single major deployment. The patent was granted. The organism worked. The need, as the Exxon Valdez and later the Deepwater Horizon demonstrated, was real and enormous. The Deepwater Horizon disaster alone caused an estimated $61 billion in economic damage. The International Tanker Owners Pollution Federation estimates roughly 6,000 tons of oil enter the world's oceans every year from shipping accidents alone, before accounting for pipeline ruptures, blowouts, and chronic infrastructure leakage. The bacteria that could clean it up ask for nothing except nutrients and time. So why hasn't it been used? Start with regulation. Releasing a genetically engineered organism into an open natural environment, the ocean, a coastline, a river delta, requires EPA approval under the Toxic Substances Control Act. The agency must be satisfied the organism will not outcompete native species, will not transfer its engineered genes to other organisms through horizontal gene transfer, and will not persist indefinitely after the oil is gone. These are not unreasonable concerns. An organism engineered to outcompete everything in nature at consuming a particular resource carries competitive advantages that natural selection did not produce. Introducing it into a complex ecosystem is an experiment with unpredictable outcomes. One elegant modern solution, discussed in the context of Chakrabarty's organism, is to engineer a suicide switch into the bacterium, a genetic mechanism that causes it to die once the hydrocarbons it feeds on are depleted. Without food, without persistence, the organism cleans the spill and disappears. The suicide switch matters for a reason beyond safety. It is the specific mechanism that answers the deepest concern of the EPA, the risk that engineered genes spread to other organisms in the environment. A bacterium that cannot survive past its task cannot spread what it carries. The suicide switch does not just make the organism safer, it makes it approvable. It is the difference between a biohazard and a temporary tool. In 1971, that engineering was beyond reach. Today, it is not. CRISPR-based tools have made programmable kill switches demonstrable in laboratory settings. The mechanism exists in principle. But the switch has never been developed into a field deployable version of Pseudomonas putida, not because the science forbids it, but because no commercial pathway has been built to take it there. Even setting regulation aside, there is a second problem in the oil itself. Pseudomonas putida degrades roughly 2/3 of the hydrocarbons in a typical spill. The remaining third, the heavy polycyclic aromatic fraction, it cannot touch. These are also the most toxic compounds. A cleanup tool that handles 2/3 of the problem and leaves the most dangerous third untouched is useful but incomplete. And even a perfected organism would face a third constraint: deployment context. Bacteria grow fastest in warm, nutrient-rich, well-oxygenated water. That same disaster occurred at 1,500 meters depth, where the water temperature was 5 degrees Celsius and oxygen was limited. Natural bacteria adapted to those cold, deep conditions did eventually bloom and degrade significant portions of the plume. Chakrabarty's organism was built for surface spills, the Exxon Valdez, not the deep water horizon. In the 50 years since its creation, nobody has built the deep water version, not because it cannot be done, but because the path was never cleared. None of these barriers are insurmountable. They are engineering problems, regulatory problems, problems of investment and will. They are not fundamental scientific obstacles. But there is a fourth barrier that the other three obscure. Cleaning up oil spills is not a profitable business. The companies that extract oil are liable for cleanup costs, but liability is managed through insurance, lobbying and settlement, not through investment in better cleanup technology. The agriculture industry, the pharmaceutical industry, the gene therapy industry, all of them had direct commercial incentives to push through the regulatory and legal obstacles to engineered organisms. They funded the research, they built the supply chains, they wrote the standards. Nobody profits from a cleaner ocean. And so the suicide switch remains unbuilt, the regulatory pathway remains unopened, and the bacterium remains on the shelf. Ananda Chakrabarty spent the rest of his career at the University of Illinois at Chicago, shifting from environmental microbiology to cancer research, founding biotech companies, advising the United Nations. He died on July 10, 2020, at 82. The organism that reshaped an industry worth hundreds of billions of dollars has cleaned up no oil spills. The next major spill could be met not just with booms and dispersants and 11,000 workers, but with something invisible and voracious and precisely calibrated to the problem. The science is not the obstacle, it never was. The bacterium has been ready since 1971.